So, hey everyone, welcome today to the webinar. I hope you all are settling into your new work from home routines today and appreciate you taking the time to be here. We're really happy that we could still go ahead with this webinar. Joep has been very busy, so thank you very much for taking the time out to deliver this content to us all. So my name's Megan. I'm training coordinator here at NESI, and our webinar topic today is workflow languages, specifically common workflow language. We have Joep de Ligt from ESR here to explain the benefits of implementing genomics workflows and to demonstrate what a CWL workflow looks like. So, but before we get into all of that, I just have a few standard housekeeping items to cover. We have quite a few people in the webinar today, so I do ask that you keep yourself on mute if you are not asking a question. I think there will be some points in the webinar where Joep will ask if anyone has questions. If we're at one of those points, feel free to unmute yourself. Otherwise, you can post or type your questions into the hackpad that we have set up. And I am going to see if Joep can share his screen for a second to show us how best we should record those questions in the hackpad. Would you mind doing that for us, Joep?
I will just have to drop out for Zoom for 1 second to allow it to do this, and we're just going to do that for 1 second.
Sure. Okay, so while Joep is sorting that out, I will just continue on to say that he's going to present for 30-40 minutes or so, and then we'll have a discussion section after where we can have more general discussion on workflow, other workflow languages, what you guys prefer, and other questions can be addressed. And the webinar will be recorded. It is being recorded right now and will be posted to NESI website. If you signed up for this webinar, you will get an email once that information is uploaded, once the content's uploaded. So if you don't want to accidentally, are you back? Yep.
Yep, I should be able to share.
Awesome.
If you stop sharing.
Yep.
Yep. There we go. So I'm just sharing my browser with you. So if you follow the link to the hackpad, you should see something like this, or in the top, you can change your view. So if you just want to see the content, you can do like that. And you can see here that there's one comment at the moment. That's one that I just created a moment ago. And you should be able to go in there and see their comments. So this was my test comment. So you can see that I'm logged in as a guest now. If you do have a HackMD account, feel free to use it. If you don't want to use that, you can do it anonymously. You can type your message like that, and we should be getting an overview of the different comments that people are making. And if you don't want to see them, you can just click it away like that. So that should allow us to collect your questions in the same format as where we have our content for today. Any questions from anyone about that?
Yeah, and you can also, if you have little random chatter things that you would like to mention, you can use the chat. I'll be monitoring that as well, the Zoom chat, I mean. Cool. I think that I just reposted the hackpad link as well in the chat. If you came in a bit late, it's in there. And yeah, if you're having trouble with that, just let us know in the chat. I think that's it. I don't know if I said this before, but if you don't want to accidentally be recorded in the recording of the webinar, make sure your video is turned off. At the bottom left of your Zoom screen, there is a stop video button. So make sure you click that if you don't want to be seen in the recording. With that, I will pass things over to Joep.
I just saw a question from Dinidi if we do need to be logged in, or?
No, it's just you.
Okay, that's okay.
Yeah.
So you should all be able to see my browser for the HackPad. I'm just in view mode. If you don't, please raise your hands, and then Megan will probably let me know that there's something up. So yeah, as Megan illustrated, we'll be talking mostly about CWL, but I would like to start off with a message that it's not that you have to use CWL. It's just that that's the language I think is most useful. I know that there's some people that really like Nextflow or SnakeMake, and that's fine. Maybe in times of self-isolation, my usual analogy doesn't quite work, but I'll give it a go anyway. It's a bit like sex education in high school, where you teach people to use a condom, but you don't really care whether they use one with a particular flavor or with ribs or whatever. You want them to be safe, right? The same thing goes sort of for workflows. It's about that if you're using one, you'll already be doing a lot better. And that's not just in terms of reproducible science, but also in terms of helping yourself be effective, right? So I brought a whole bunch of bananas today. I will be showing 2 flavors of condoms. And that's just so that you get an idea of how that would look like running them on Nessie, so that you can get started with it after the end of this today. But there's others out there, and they all have their different flavors, pros and cons. And we can discuss some of that if you want at the end, if there's a lot of people that would like to have that discussion. But my biggest take-home message is at least look into one of them. And if your group is using any of the ones that you'll see today, go for that one, because then you'll have support. And that's where we start. So we'll be going through some slides later on, but the gist of it is that we are here to help our future selves. So this is a comment from XKCD, where he left comments in his code for his future self, because at the time that you were writing it, you knew that you would have to go back to that piece of code. And with a lot of things that we do in research, we have to go back to that one particular step. So for example, in genomics, read mapping against a reference genome is something that we do in an insane amount of times. And it's a bit silly to write the piece of code to do that every single time. That's why there's tools like BWA or stuff like that that do that, right? But like there's a tool, you can also have that for yourself, for a piece of research that you are doing, a piece of code that you have to reuse. So we'll just spend a bit of time talking about how we can help our future selves when it comes to tools that are not standardized or published in a way. And that is that your coding should be dry. We can leave the wet stuff for the wet lab, but dry stands for do not repeat yourself. Or as Bill Gates puts it, I rather hire a lazy programmer, because they will make sure that they do it once and that then they don't have to worry about it. So it's about saving yourself the effort and time of rewriting stuff, but it's also about having your code or your work in such a way that you know what it's going on and what it's doing. And writing functions is a great way to do that. So here's an example from something in Go. And there's a very simple function definition that says repeat X times. And we are going to repeat a string for a number of times, which is a number in an integer in this case. And as arguments, it takes a string and an integer. So which tells us that this function is going to repeat a piece of text a number of times. And it will then print that to the screen. What this function does really well, it has a clear purpose. There's a clear output of what it does. And it's testable, right? So if you want to test this function, you can type in a string and say that it has to repeat it 5 times and see if it does that. That's another thing that, especially in more complex pieces of code, becomes really important, in the sense that you want to be sure that it's doing the thing that you expect it to do. In programming, there's lovely frameworks and things that you can do, like unit testing or continuous integration. There's all the nice IT buzzwords, but the most important thing is that you want to be sure that it's doing what you expect it to do. Just like in any decent scientific experiment, we have positive and negative controls. You also want to be able to do that for your functions. So that's just a philosophy that you can embrace in your coding that will help you to be more effective, but also to be more reproducible. Because if you can define what a function should do, someone else can then reproduce whether that is actually what the function does. In pipelines, so when you are not talking just about one piece of code, but many pieces of code, this means embracing workflow languages. Because a workflow language defines a number of steps that you want to do. And for each step, it defines what program is gonna run that step. Which means that for this example that you see here, that there's 3 sort of modules here. One is for alignment, one is for variant calling, and one is for sort of piping, maintenance, household stuff. That means that if we have this workflow that we call alignment, we can use that in many different projects where we have to do an alignment. So that means that everyone that wants to do a particular read mapping to a reference genome could use that workflow. It also means that if you have different tools that do a similar step, you can compare them. For example, you could have one BWA alignment step, one minimap alignment step, and another, your favorite aligner alignment step. You could have them as workflows, and then you can feed the same data into all 3 of them and get the data out at the end and compare those alignment steps. And then if you have a bigger workflow, you should be able to interchange those pieces. So what this gets you is that if you have a new aligner, you can change or make a new workflow and just plug it in. Well, what that just plugging it in looks like is something we'll explore later. But that's the general concept of it, that you have these steps that have a discrete input. So reads, they need some sort of parameter, your reference genome, and then they perform a step that will result in a aligned file, so a BAM file or a SAM file, depending on which one you use. But that's the concept of a workflow. There's this clearly defined input and output, and what happens in the middle is you preferably care about as least as possible. So it's about defining what you need to happen and having the tools or pipelines that can do that, but that can easily be swapped out for newer tools or other tools. Because when you're doing your research, you care about what comes out at the end, which is something that you can do a biological interpretation about, or some new and exciting piece of information about how those tools perform, or what they might miss in terms of their sensitivity or specificity. You don't necessarily want to care about the underlying nitty-gritty details. At Nessie, you're slightly spoiled with all these lovely people installing software tools for you, but you might be in a situation where that's not as easy. For example, if you end up in a cloud environment or in an infrastructure where all those tools are not pre-installed. So what do you do then? So at this point, I'm going to switch over to some of the slides that I also presented at eResearch to talk a bit more about the philosophy of workflows. But I just wanted to check with the room at large if there's anything in the bits that I've said thus far that you think like, wow, what is that about? Or could you elaborate on that a bit more?
And feel free to type in the chat or type hand in the chat to indicate that you have a question, so we know to pause for you.
I have a real quick question if I can.
Sure.
I understand testing and having known outputs, but when it comes to some of these genomic pipelines, you just have a small dummy file that you use that has a known output that you're testing against to make sure all the stuff is still functioning, or what are we doing on pipelines like this, as opposed to things like repeating text strings, which are easy to store a known outcome for?
Yeah, no, very good question. So this, of course, depends on how robust you want it to be. Most tools these days come with what they call a minimal test set. And that's just really to test if it's doing what it's supposed to be doing. So that's a good practice to include a minimal piece of representative data that you can use to run quickly through your code and see if it's doing what it's supposed to be doing. With some of the processes that we use in genomics, there are, of course, random number generators or other less reproducible things involved. So it's about having a test set that reflects the biological signal that you want to get out at the end. So if you're talking about an alignment and variant calling set, there's things like the genome in a bottle if you're working with humans, but other well-published genomes if you're working with a different species, that can give you a set where you evaluate the performance or the actual accuracy of the pipeline based on a sort of silver or gold standard of data. So that's how you typically evaluate these types of pipelines.
Any other questions or comments for the moment? Cool. I think that's it for now. Yep.
Excellent. So this should theoretically work. There we go. Boom. So in the hackpad, you can find the actual links that I'm using. It's all available online. I typically publish my stuff as a CC BY, so feel free to repurpose it anywhere else as long as you allow other people to then repurpose your stuff. I'm all good with that. So we also published a protocol in that same license that we called Scalable Workflows and Reproducible Data Analysis for Genomics. I'll be using bits of that protocol in the demo session later. But what that does is that there's different people that have their own favorite workflow language. We got them to build that in a sort of teaching material style. So you can go to that protocol and check that out. There's different people from different backgrounds involved in that. My disclaimer here is that the creator of CWL is on that protocol, and that I am arguably a bit of a CWL fanboy. So I might not be completely impartial, but there's also SnakeMake and Nextflow and Geeks workflow language in there. So I think there's a decent representation of this sort of major workflow platforms out there in that protocol. And for me, a reproducible workflow, you can compare it a bit to if you're going to be cooking. So there's data in cooking that would be your ingredients. There's tools, so there's your appliances, and there's settings like pounds or grams. When you're talking about reproducible, it just doesn't become about does the tool do what I want it to do, but it also becomes about, is it useful for other people, and how can they use that workflow to either reproduce my data or reuse it, which is where the real beef comes in for science. So preferably you have some standards like pounds or grams. You might want to agree that you're all going to be using the same thing. So that's what workflow languages. There's also some of that debate going on. The good thing is that we have containers these days, and all of these workflow languages support containers, and you'll see an example of that later in the demo. So that's one of the standards in terms of the tools, the appliances that we use in our science. That's starting to come. And some journals are now even requiring people that if you're publishing a piece of software, that you have to provide a container so that people can grab and run it with minimal hassle. Data, that's about formats. So one of the things that you do in a workflow language is that you specify the format that the data is that has to come in. So an alignment will always ingest either a FASTA or a FASTQ file. Right? Of course, for people with nanopore data, they can go like, yeah, but I want to align my FAST5 data. Well, that's fine. But then we need to define a new workflow or a new tool that can actually ingest that data and do the alignment in a squiggle space. That's fine. And that could still result in a BAM file in the end, but it might have a different input. So it's really important in a workflow that you define those inputs and outputs, and that you are very specific about what you expect to be coming in. And then the settings, workflows typically take a settings file. So of course your tools will have parameters, and you might have a specific parameter that is different from everyone else. You want to log that typically in a file where you specify which particular setting that you will be using. Just like the temperature in your cooking, that might differ if you're at a different altitude. So those settings might be different if you're in a different compute environment. So this is an example from EBI. They have made all their metagenomics analysis available in common workflow language. And there's a nice little tool that you can view workflows with, made by the consortium. And what you see here is that there's different inputs. So those are at the top in the sort of dotted line box. So there's a different model and there's a sequence going in. Then there is a parameter that is being set that is in the sort of purplish color. That's the 100,000 number. And they fit into the split FASTA by number of records. So what that tells you is that these sequences, they're going to be split into chunks of 100,000. Those chunks then go into a gene scan where there's a certain model and some other sequences going in. And that then results in predicted nucleotides and amino acids. And that is then what is the output. And that is at the bottom of the workflow here, again in blue. So those are typically your files or your actual data points, and everything in yellow is your software bit. So this provides us with a very descriptive way of seeing what a workflow does. The good thing is that if people make their workflows available in CBDL like that, you can have this sort of overview of a pipeline without even touching it, right? You only have to view it, and you can see if the steps that are going into it are useful to you and are sensible, right? You don't have to spend hours of digging through Perl code or Bash scripts to see what people might be up to. There's a nice structured high-level overview of the steps that they are taking in their analysis, which I think is pretty neat because I'm a lazy man and I just want to know if it works for me or if I need to keep looking for something else. I will skip this part. That's more about the scalable and portable part. If you are ever going to be in a cloud environment, feel free to look into that. But since this is about Nessie, I'm just going to skip those and acknowledge the people that were involved in putting some of that together. Now I should be able to go back completely. Here, There we go. So that's as far as sort of my introduction to workflows goes. This is a very high-level introduction. At the top of the document, you will see that I've linked useful resources. There's a very, very good tutorial made by the University of Melbourne, where they go much, much more in depth into how you could start working on these workflows and how you can start building them yourself or how you can start adapting them. And there's the links to the slides and the protocol that I was talking about as well, if you would want to look into that. If you feel that that is something that you want to go into, I think it's a good place to start, either at that protocol or their tutorial, to just get a bit of a better idea of how that would work for you. So this is where I have the demo time title going in. Before we do that, there is one piece of software that I would like to show that I have found incredibly useful for people that are starting out with building their own workflows. And that would mean that I have to now switch over to this tool. So I'm gonna change my screen share. You should now see the Radix Composer. This is a piece of free software that has been made by 7 Bridges. And what they have built is a visual interface to interact and build workflows. But you build them in such a way that there will be a CWL, so a workflow language file, coming out at the end of it, which is awesome because, as you will see in a second, we can run those on Nessie, which I think is cool. In the left-hand panel, you will see some of the workflows that I have git cloned. You will see those git clone commands in the hack pad so that you know where I'm getting them from. And if I missed one, my apologies, then just shout out and I'll add it. These are different repositories that people are hosting on GitHub that have a whole bunch of CWL definitions for tools preloaded. So for example, there's the bio CWL tools. There's CWL definitions for GTK, Callisto, Star, BAM tools, BWA, Kraken, all kinds of nice bio tools. So since this is a genomics session, I thought I'd show you people that. If you open one of those, you might see something like this. So this is an RNA-seq alignment workflow. So there's Star in the middle, which is for those people that do RNA-seq, is sort of the read counting and quantification method for a lot of standard RNA-seq experiments. And you can see that it also includes the generate genome step. So even if you're using a genome for which there's not a star index, you can use this pipeline. You can see on the left which inputs are there, and you can map that to files that might be on your system if you want to give it a go. And you can see what comes out at the end. I just have to minimize this so that there's splice junctions, chimeric junctions, and all those kind of things going in. And you can even see here the different outputs. And you can see the settings that are going into this tool. So at the moment, there's no genome file specified, but you could specify that in that interface. If you are more into very complex workflows, there is also a group of people that have analysis workflows for human genomics specifically, and they have increasingly complex pipelines. So there's ChIP-seq, there's bisulfite, there's a complete RNA-seq pipeline that goes from reads to mapping, there's somatic exome, there's tumor. And if you open up one of those, you can see that there's a lot more going in. So this is the chipsec pipeline from them. And you can see that there you have a whole genome sequencing QC workflow that has all kinds of metrics like GC bias, per base metrics, coverage on target regions. And then there's a module that goes from your raw sequence data to BQSR files. So there's a whole suite of tools that they have mapped and integrated that you could start using. And if you don't want to use it, you could even just check which settings they're using. And all of these things are nice because you can have a look at them and have a play around with them. And you can also add things in. So for example, if I would want to add a specific step into this, so we have, for example, a BAM to SAM file here. So if we have a BAM file, we might be able to do variant calling. So we could have GA2K going in here. So we might want to input a base recalibrator, and it will tell us what it will need to have as inputs. And I'll just zoom in a bit. So that needs a reference file. Well, probably we have some sort of a reference file in here somewhere. There we go. So we could drag that and point it to the reference file for our base recalibrator. What this will do is that if you run this workflow and you specify a reference, that one reference file will be fed to your QC workflow, to your raw sequence to BQSR, and now also to your base recalibrator workflow. The thing that comes out at the end is a valid CWL file. So you can then share that or run that wherever you want. So you can start to build fairly complex workflows quite easily without necessarily having to code CWL yourself, which is good for people that want to get work done, but might not be as comfortable writing all of that themselves. So that's just a piece of software. I have no stocks or anything in this company. I think that this is one of the more useful things that they have ever produced. And feel free to have a look at it if you want to get into building workflows or exploring them. I have to stop sharing and then go back to my demo. So I'll be showing in a couple of minutes also how that actually looks like on Nessie in the terminal. But there's a couple of things that I did as a prep that if you would want to reproduce this, you could. This should all work on Nessie. I ran it yesterday and it worked. So fairly certain that unless stuff has changed overnight, it should. So here's some of the things that I've downloaded. So I've downloaded a small data set. So this is from the Melbourne group. They have a nice little data set that we can use. And I've also downloaded some general workflows that are provided by the Common Workflow language. And I've downloaded the protocol that is the one that I presented earlier that shows some genomics analysis in different workflow languages. We have to prep our Nessie environment. So I've done some module loads for SnakeMake, CWL2, and Clustal Omega. Dinny told me that they've been updating the cluster one. I've not updated that. I can see that here. It should have been there, but anyway. You should be able to not specify the version, but I thought I was going to be good and be very precise about the version that I was using. So if we want to use a predefined tool, here is an example of a BWN definition of a CWL file. So this is what the actual file looks like under the hood. So there's a container specified here. And this is where the power comes in if you are in a different environment than Nessie. If you run this on your own computer, it will use the exact same software as it will use on Nessie because it gets a container. It says here Docker pool, and you might go, well, but Nessie doesn't allow Docker. Well, as you can see, we can also do that with Singularity. This is just pointing to where those containers are, and if there is a Singularity container, you can use that on any system that only has Singularity just as well. This is telling us that there's going to be a bunch of files and that there's going to be of type FASTA. Now, we could have typed that in with just letters, but that's not very specific. So what they've done here is there's actually an ontology describing different file formats, and they reference specifically that format, which then tells the system that that is a FASTA. So you can see that this is a very precise and reproducible typing of the input. Then there's some files that it's going to be making in the index. So the index files that we'll be using. And then there's some optional arguments about the number of threads, seed lengths, and all those kind of things. So you can see that someone has gone through a lot of trouble of basically documenting what BWA does and what parameters it has for this specific version, because as we all know, parameters change over versions. So if we would want to run that, we could pop into Nessie and we could run this. So we've loaded CWL tool, so that's a tool that can run workflows. We're going to specify that we are going to be running it using Singularity like this, that we are going to run this particular workflow, which is in one of the repos that I just cloned into my workspace. This is the data from Melbourne. So if you follow the above commands, it should be in the same data structure, but there's a R1 and an R2 reads. There's a reference file that we're going to be using, and then we're going to have some sort of output. Now remember that we didn't tell it how to run BWA at all. We are just using default settings here. We could, of course, change that, but this should then work. So I will have to stop sharing this and then share my terminal. And there will be a bit of back and forth in this, just bear with me. So here we are on Nessie. I am in the GA teaching directory where I've made a workflow workshop folder. So here we can see the data in core. I'll make this a bit bigger so that everyone can read it. There's the core. That's the data set that we downloaded. This is the scalability and reproducibility chapter, and this is the workflows that we'll be using. And just do not have to go back to the presentation. I'll just grab it from here. So this is the command that we were going to run. And I figured out that should work. So I just have the date in front of my line here. We haven't loaded the modules yet. Very good. Module load CWL2. There you go. So what this will do is it will start the workflow, and it will look it will look for the files, and because I didn't specify the reads a second time, as I should have. Now it's telling me that I haven't loaded Singularity yet. This is what you get if you don't follow your own instructions, people. Always follow your own instructions. So what you can see that it went and collected the data. It provides us with some logging about orientation, percentile, And it also provides us with the information of the job that we have just run. So it took a couple of seconds to complete because this is toy data. But there's a file that it has created with a certain checksum. So you can even check the integrity of your workflow right away. And that there's the location where that file has ended up. And that's the final process status is success, right? So what this does is it takes a workflow that we have just taken from Git. It takes a data set that we've gotten from somewhere that provides useful test data, and we have run it with using the cwl ---- singularity command, just like that. Because it grabs the container, this should work the exact same way everywhere. And it has these checksums so that you can check if the actual data is exactly the same in terms of the output, which for this one should work. So then this is the same output as we've just gotten. So you can see that it's sort of taken the same time, and it should be the exact same checksum value as we just had, which is good. That is using one tool. What if we want to use a workflow? So this is where I'll show a SnakeMake example, just to show that I am not only a CWL person. I can also have love for SnakeMake. This is a workflow that has different steps in it. So it's just not just one tool that you saw, but it has a couple of tools that work in order to produce a result. So there's a cluster step here, there's a PEL to NEL step here, and there's a CodeML, which is basically the last bit of output. What this does is that it is a gathering step here of all the data, and I'll show you in a minute why that works. But it tells the system that it's going to run this for a whole bunch of files, and then it's going to run all of these steps. And I will have to module load my SnakeMake and other things. So I'm just going to copy that over without the version because I know that it might have changed. And pop back to the terminal. So we need to load SnakeMake. And we need to load cluster omega. There we go. So if we then go into the scalability and reproducibility chapter, there's the different workflow languages. So you can see that there's CWL, Nextflow, SnakeMake, and the Greek Geeks workflow language. The actual Docker containers are provided there as well, but we're just going to pull them from global. And there's data. And if you look in data, there's a whole bunch of these clusters. They all contain some sequences. So for example, here we can see that there is the amino acid sequence and the nucleotide sequence. So this relates to the same sort of thing that you would have seen from EBI just now. And this is something that we want to do on a whole bunch of samples, files. We want to do the exact same thing. And that is a 3-step workflow. So if we go into SnakeMake, we can see that there is a snake file here. That is the file that you have just seen in the hackpad. So you can see that there's the gathering of samples. So there's a range from 1 to 73. You could also use a wildcard if you want, but this is about reproducibility. So we don't want the pipeline to be dependent if more clusters show up. So it's just clusters 1 to 73. And we should then just be able to say snakemake. And it will start running. So this is just the cluster step. So it's going through all the different files you can see here. And then there's some bits in the end that will error out. And we can go into details why that happened in the end if we want. But that is a predefined workflow that someone has put together, not knowing anything about the existence of Nessie, that we can use a Git pull to get that workflow and some of their test data and see what it does. And of course, we could point it to other data by ourselves, and that would work just as well. In the notes, you will see that there's also the example of how to run that with CWL. That would look like this. In the CWL implementation, they chose to have a parameter to point to the data rather than include that in the workflow, which is, I think, slightly neater. But that's a matter of taste more than anything. It's not dependent on the language. Where you can, again, use CWL to -- singularity because it's not allowing Docker. Point to the workflow and specify the data that you're going to be using. Now, the thing to note is that for CWL, we have to specify the singularity and Docker containers. Snakemake didn't use containers because the tools are actually available on Nessie. So Clustal, we just loaded that through the module, and it can use the native version of those tools. CWL can do that, but it has the sort of style recommendation that you use a container to do it, because it's more about the reproducibility side of things. But that means that if you have all your tools available, you can start with a snakemake pipeline a bit more easily on Nessie than with a CWL pipeline. So if we then talk the last couple of minutes before we have some questions and discussion, there's a lot out there. I've just linked a couple of things here. I think embracing these workflows allows you to truly stand on the shoulders of really, really tall people. I refrained from the more fantasy equivalent that people often use, but also some truly awesome groups. So within the human genomics space, there are whole consortia dedicating their lives basically to making curated workflows for RNA-seq, for exome sequencing. The same goes for the ENA about metagenomics, the stuff that they do there. They are one of the leading standards in that. You can use their workflows by just cloning their Git repo and start running them. So I think that you are helping yourself by embracing workflows. And as a bonus, you will get very nicely reproducible and reusable pipelines at the end of it, so that when you publish, other people can use your awesome stuff much more easily as well. And that is where I would like to end off.
Thanks so much, Joep. That was really awesome information. And we are going to open up the floor to some questions and comments. Before we do that, though, I'm just going to share a link to a very quick survey because it, of course, takes time to learn how to implement workflows for your own research. And we'd love to understand what support and training you guys prefer. So if you take a couple of minutes while we're having this discussion to fill out that survey, that would help Nessie, Genomics Aotearoa, ESR figure out how we can collaborate to offer more things like this to help you guys learn workflow stuff. So with that, does anyone have some questions or comments? And I'll just remind you, you can put hand in the chat to indicate you have something, or else unmute yourself. And if we have a few people talking over each other, we'll just figure it out on the fly.
Thank you for that. I have a quick question. So we have, for example, from Broad Institute, the JDK best practice workflow. They are sort of doing their own little thing. Is that also somewhere available in CWL or something else?
Yeah, so the Broad has chosen to invent their own workflow language. They call it WDL. So they have their best practices available in that format. A couple of groups have taken it up onto themselves to transform those into CWL workflows. There's a couple of different style changes that need to happen, but sort of the most important things like the default settings and the steps to include are there. So there are groups that are doing that exact thing. The good thing is that the Broad seems to be getting around to supporting some of the other languages as well, CWL being the first one that they are going to allow to run on their sort of systems. With these things, we will see what sort of the community preferred standard will be. The one positive thing that I see about CWL is that SnakeMake can even run CWL languages since a little while. The Broad is picking that up. So in terms of sharing pipelines globally, that seems to be the preferred standard. But for working locally, you can leverage off of those other people that are doing those sort of translations, or you can just decide to run WDL natively.
Thanks.
Hello.
Hello. Is that another question?
My name is Veena. Hello, am I audible?
Yep, I can hear you.
Yeah. I'm sorry. Yeah. So, hi. This is an interesting talk. Thank you. So the question is regarding including a code snippet written by us. So it's like I'm just asking a question regarding a workflow where we have a custom script made by me, and there is a tool, something like BWA. So so if we have to put this script into this CWL with the Rabix composer. So in that case, it like we have to write our own code snippet to include that particular code into Rabix.
Yep. So if you want to do that, I really recommend checking out the tutorial from the Melbourne guys. They did a really good job at introducing people to how to write your own CWL. What it comes down to is that in CWL, you will have to define what the input of your tool is and what the output of the tool is and what parameters there are. That will instantly give you your documentation as well. So which is useful anyway. So that's the bit that you would have to do. If you're sort of doing it for the first time, it will probably take you a couple of hours. But yes, as you get into these kind of things, that gets more easy, and it's not necessarily a thing that you only have to do for it to be in a workflow language. It's also just useful documentation in general. And if you then do it in a format that describes your tool to others, I think that's a good thing.
Okay, so basically this individual script as well as the tool will come in one single .cwl.
So the CWL is the description of the tool. The tool itself will just stay where it is. You don't have to package that.
Okay, thanks. Yeah, thank you.
Cool. I think we have a couple of questions in the chat. So is the drag and drop workflow designer shown on the last site functional? Or wouldn't it be nice if that was functional?
So the drag and drop thing that I showed in the last slide, that is the Rabix composer. So it's just a GIF showing that, just to remind people that there's a visual interface to building workflows, and that is functional and freely available to everyone that wants to use it.
Awesome. And where can we get the analysis workflows?
So in the Hackpad, there are the different repositories that I've used. So the one that has the analysis workflow is from the human genomics side. So they have this quite comprehensive suite of tools where they have complete pipelines, if you will. This is just a snapshot of what is out there. So if you start looking for CWL pipelines of your field of interest, whether that's metagenomics or whether that's human genomics or something else, there's more and more coming online. And I just wanted to highlight these because there are a group of people that are doing quite a good job at curating them. So I know that I can just drag and drop them and they will work.
And then I think there's a hand from Peter.
Yeah, I was wondering if you could talk about integration with Slurm, because it seems to me that on an HPC system, that's the novel and possibly difficult aspects. There are two ways you can do this, right? You can put the whole workflow inside a single Slurm job, or better, I would argue, you can try and dissuade that workflow tool to use Slurm as a back end, but that has some complexities. So could you talk about that, especially with relation to CWL?
Yeah, no, sure. So SnakeMake has some of that functionality built in. The CWL community took a slightly different philosophical approach in the sense that they think that workflow should be completely separated from the architecture. So what has happened within the CWL community is that there are several runners that can do that. So the one that is most often used is Toil. It is just like CWL2, but it has bindings to Slurm. So if there's something that can be parallelized, it will make a nice array job out of that for Slurm. So if that is something that on Nessie people start to want, of course, that is something that could be implemented and could be explored. But that's sort of a philosophical choice that has been made, that that part should be within the runner and not within the language.
Okay, so we are at 1 o'clock now, so I'm going to say that that is all we have time for today. Thank you, Joep, so much. That was really, really informative and helpful. And the recording and the Hackpad and everything will be sent to you guys, posted on the Nessie website. So keep an eye out for that, but have a great afternoon, everyone. Thanks for coming.